Um, for those of you who haven't met me, I think I've met most everybody before, but my name is Brian Murphy, and I'm an associate professor at the University of Illinois Chicago. And the purpose of this presentation is to kind of get us all on the same page and give everybody an update as to the big grant that we all just received. So a huge thank you goes out to all of you. You have been amazing collaborators so far, and because you have been so timely in contributing to both the writing and contributing resources to this, we were able to put together a nice grant. And as you all know by now, it was funded by our National Institutes of Health. So it's classified as a D43 grant, and the project period will be from around July of this year. We're about to receive the funding, and we'll work with your institutions to transfer you the money. And it will end around 2022. Now, this is a 5-year grant, but there will be an opportunity somewhere in the middle to renew this for an additional 5 years. So the title of the grant is Enhancing Basic and Translational TB Research in Northern Vietnam, and it's going to be a collaboration between UIC, the Vietnam Academy of Science and Technology, and the Institute of Marine Biochemistry there, and the National Institutes of Hygiene and Epidemiology. And before I get into the grants and I just want to give a brief introduction of the history of collaboration between UIC and VAST, because it goes way back, way before my time. Professor Dol Soeharto and Van Hung Nguyen received MacArthur Foundation funding and NIH ICBG, which is International Cooperative Biodiversity Groups funding, and basically they focused on discovering new drug leads from plants collected in Vietnam and Laos. And this collaboration was a long collaboration, and it was incredibly successful with a lot of peer-reviewed papers, patents, PhD dissertations. Now, Kung was kind enough to extend an invitation to me to come, and he was a great host at IMBC. So I took a trip there in 2011, and we started a collaboration that has existed to this very day. And as you can see on the left, we've been very successful in producing papers between both of our universities, and we have a nice timeline in which we obtained a small amount of funding in 2011. We did a student exchange for a little while. Now Dr. Cuyen Vu, she graduated from IMBC, and now we can continue our collaboration on to about the 10-year mark with this NIH funding. So of course the three institutions involved, as you guys know, you are in Hanoi, and Chicago is located in the middle of the U.S. This little dot right here on one of the Great Lakes, this is Lake Michigan. And I put this down really for my use. But this is the distance between, NIHE and VAST approximately, and I'll be needing that in the next couple of years. So the overall purpose of our grant, what we are going to do as mentors, is to train young scientists in TB drug discovery, and between us at both universities, at all three institutions, I think we have an incredible degree of knowledge, and if we combine all of our powers, we're going to be able to set up a really nice multidisciplinary drug discovery program, microbial drug discovery, that in particular focuses on TB, and the NIH was really interested in funding TB. So alright, let's just dive right into it. The three aims of our grant are as follows. Aim 1, the NIH expects us to establish 3 independent investigators, 2 at IMBC, 1 at NIHE, and each of these 3 trainees will receive 2 years of training at UIC in their specific discipline. And I again thank all of you for reaching up to your administrators and higher ups to help create these positions and help select these candidates. Aim number 2, we're going to provide short and long-term antibiotic discovery training to a number of different predoctoral students, 8 at IMBC and 1 at NIHE. And aim 3 is really going to focus on NIH, and we're going to they have an incredible program that exists there already, and what we're going to do is just simply try to enhance that by adding biological assay research infrastructure that focuses on a bunch of in vitro MTB assays, but also, if the program is extended, hopefully in vivo MTB assays. And that's going to be through collaboration with UIC's Institute for Tuberculosis Research, led by Dr. Scott Franzblau. Now, the administrative structure of our program is as follows. We have a training advisory committee that is led by Dr. Dol Soharto, and on the committee is Van Hung Nguyen and Leti Quinn Mai from the NIH. Now, the three PIs are myself, Kung, and Dr. Ayn Dudang. And there's a mentor team. There's a mentor team at UIC, IMBC, and NIH. And all of us will be providing training to the postdoctoral and predoctoral trainees. So here are the trainee teams. Scott Franzblau and Sang-Hyun Cho are both at the Institute for Tuberculosis Research, and so they are TB biologists. Richard Van Bremen and Laura Sanchez are experts in mass spectrometry and natural products, and Shura Mankin and Alessandro Eustachio are experts in molecular biology, genome mining for natural products, and antibiotic mechanism of action. Now, the mentors at IMBC are Kung Hong T. Mai Don and Hai Dang Nguyen, and at NIH we have Ayn Dudang and Ayn T. Van Nguyen. I'm sorry, I'm working on my pronunciation, so I apologize if I messed any of the names up. But correct me when you see me, please. So this is our all-star team of researchers and mentors that we will all be working together to train young scientists. Now, we're going to be training them based off of this drug discovery pipeline that I'm sure you're all familiar with. You have sample collection from the environment in some way, and because we're focusing on bacteria, we'll be collecting samples from the environment, growing bacteria, and creating microbial libraries, and then libraries of small molecules from these microbes. We'll screen them against different biological assays, in vitro TB assays, dereplicate leads, so basically figure out which projects are most likely to produce completely new compounds, and then identify the leads through a number of different spectroscopic means. In years 1 to 5, this is kind of the overview of our program. So on the bottom we have our Vietnamese institutions, and on the top in red are the UIC institutions. So postdoctoral trainees will be sent to UIC, and in return we do 2 years of training at UIC, and then upon their return to IMBC and NIH, the mentors there will be picking up that training and continuing it, and you guys will be incredibly important for their transition into their, as you know, their independent careers. But really, we'll all be working together during each of the 5 years to help train these postdoc trainees. And in the meantime, there's going to be a supply of leads for antibiotic screening to the NIH, and then a return of regular in vitro TB screening from the NIH to the IMBC. And then, of course, beyond the project period, hopefully involving more institutions. Now, I know Kung at IMBC already works with the Institute of Marine and Environmental Resources and Institutes of Oceanography, but we have some funds from the NIH to really incorporate these two institutions and do some serious like deep ocean and shallow water sampling that I'll talk about a little bit later in the presentation. Alright, so now I'd like to talk in more detail about the postdoctoral training and the transition to independent research careers. Now, this is going to take place at both all 3 institutions. Alright, so bear with me, this is a slightly complicated figure, but on the bottom we have what we've designed at UIC to be our drug discovery program, and we'd like to try and create a similar pipeline with the help of all of you mentors here. So on one hand, we have chemistry. So there's myself, who does antibiotic ID, Van Bremen and Sanchez, who work with small molecule analytics and MSD replication of small molecules. Now, a lot of this already exists at the IMBC, and we have antibiotic ID and generation, but maybe what might be useful to add is a trainee who has expertise in specific MALDI-based MS, which would be a nice complement. And that really is going to come from Dr. Sanchez, training directly in Dr. Sanchez's lab. So, MALDI is a very specific type of MS that hopefully in the long run we'll be able to implement similar MS techniques to identify microbes and dereplicate leads at the IMBC. And of course, number two, we have Professors Mencken and Eustachio doing antibiotic mechanism of action and genome mining and antibiotic supply. We'd like to train somebody directly in the Eustachio lab and partly in the Mencken labs to learn how to efficiently mine hundreds or thousands of microbial genomes for new natural products. And also, trainee 3 will be spending the majority of their time in Professor Florensblau's lab, where they will learn in detail how to set up and implement in vitro TB screening and do antibiotic mechanism of action studies. And this will complement the already existing expertise in TB research that is at the NIH. Okay, so 3 postdoctoral trainees are going to go through this 2-stage application process, and of course, Kung and Ein Dudang are going to be instrumental and very important in helping us identify lead applicants, right? So they'll be interviewed by both all 3 of us, really, and then once we select our candidates, we will hopefully allow them to interview with some mentors at UIC, just to make sure that it's a good pair, and that the pair has the same learning philosophy and that they're a good match. So after pairing, each of them will work for 2 years, and they'll begin their independent research careers after 2 years of training, with a slight transition period with all of your help. So while at UIC, these trainees are going to take a bunch of classes that are taught by specialists here. They're going to be involved in grant writing workshops, how to tell a story and how to sell a story, and hopefully I can, convince you to do a little, to do me a favor and participate with me in grant writing workshops as well, because I know that you have all been very successful in getting grants, as I've seen by your records. So hopefully you can give a lot of these trainees nice grant writing tips. And of course the grant writers are going to engage in research as well. And while they're here, design their independent research programs and continuously submitted to us for input. And I just listed here some of the potential classes. We have all of these classes are heavily focused in drug discovery, including this cool seminar course where the students really teach each other how to present. We have structural elucidation courses, advanced pharmacognosy techniques, spectroscopy courses, fundamentals in immunology and microbiology, etc. So the students will have the choice of courses here. Now, trainees will be attending these workshops in addition to lab safety courses for principal investigators, PIs, and hopefully we're going to give them some experience of managing a budget. Now, they'll work directly with their mentors, which is us on the UIC side and you on the NIH and IMBC side, to develop research plans. And hopefully data that they generate here can serve as independent data in which they can use as preliminary data to apply for research grants elsewhere. So yeah, by the end of their training, hopefully they'll be writing proposals, submitting them, and they'll need our help to do so. Now a little bit about the predoctoral training program, and this is mostly going to take place at IMBC and NIHE. So here is a summary of the 9 predoctoral trainees here. This is this top column. So in year 1, there are going to be 2 new predoctoral trainees that are paid for by the grant, for a total of 2. So in the second year, we'll add 2 more trainees for a total of 4. In the third year, we add 2 more trainees for a total of 6. And in year 4, 3 more trainees for a total of 9. And around this time, year 3 and year 4, this is where when the independent postdocs come back to start their own programs, they will be given a graduate student who can work directly under them, hopefully. And so yeah, by the end of year 5, we'll have about 9 predoctoral. And here's the postdoctoral trainee schedule. 2 in year 1, and then 1, this is the NIH candidate, will come in year 2, for a total of 3. Okay. Now, in some fashion, the predoctoral trainee's dissertation research has to involve the discovery of TB antibiotics. I've written down some examples here, whether creation of phylogenetically and chemically diverse microbial libraries, or isolation of TB leads, etc. These are just examples of dissertations, but really it'll be the mentors at IMBC and NIH that are obviously, as you've done for years now, going to decide what the projects are. But the NIH is going to require that in some fashion it involves TB research. And the postdoctoral trainees are going to serve as the bridge between UIC and Tannoy. So a little bit more about predoctoral training. There will be certain UIC graduate courses that are going to be available freely online for predoctoral trainees to download and view. Several PIs from UIC will be visiting to give short courses in person at IMBC and NIH, and so everybody can participate in those. Whoever you'd like to come can participate. But for example, you know, I will go, and Kung and I will both co-lead a workshop on sample collection, right? And we'll bring students along and teach them how to collect samples. Dr. Laura Sanchez will come and give a very detailed an in-person training session on taking mass spec data and searching global natural products social molecular networking, GNPS, this great free of charge user interface that really can quickly dereplicate natural product leads. And then maybe Alessandro Eustachio or Shura Mencken will come and give a detailed 1 or 2 day short course on genome mining of natural products. So good news is that a lot of annual funds are going to be made available to these trainees. They can use them for optional English instruction if they want, verbal and written by, if there's a local organization or private tutors, that's okay. Each one of the trainees will be able to purchase their own laptop or desktop computer of their choice. And of course, we'll be making a lot of money available to purchase software. If any of the labs need specific NMR or MS software or natural product databases, there'll be money made available for that through the NIH. Also, we have funds available for sample collection to continue the great library that IMBC has already begun to create. We have many other monies will be made available for supplies related to predoctoral dissertation theses, and a major benefit of this is the constant availability of travel funds. So for mentors and trainees, we can use these NIH funds to present our research and really promote this NIH grant. There's a little bit of funds in years 1 and 2, but there's much more funds available in year 3, 4, and 5, where we're going to be really expecting everybody to participate in both local conferences and international conferences to present their research. So that's going to be really fun to network there. And in years 1 and 2, I mentioned a sample collection plan. Kuang and I developed this plan a long time ago. He did a lot of work, so much credit goes to him here. In talking to IMER and IO in coordinating this, but you know, we've got a couple target sites that we'd like to hit, and maybe we can hit some of these in the next year depending on weather. And so this is a great opportunity for the students to be hands-on involved in sample collection. And so we'll bring these samples back and we'll create libraries. And this is going to be the start of the generation of predoctoral dissertation projects. So after sample collection, we have strain isolation, and we get to implement. Anybody who saw my seminar when I visited a couple months ago I presented this really novel way of using MALDI-MS to fingerprint microbial strains, and so that the creation of these strain libraries is not completely blind or just relying on phenotype. We have detailed MS fingerprints where we're able to tell which strains are unique and which strains are producing new molecules, so we can remove the problem of duplicate strains and duplicate chemistry in our drug discovery libraries. And then dissertation projects are really going to come from the strain libraries and from the chemical libraries that we generate. And again, here is just a little bit more detailed overview of the trainee schedule, and you can, I just put this up here for you to view. So some knowledge for us, the mentors. How do we measure success from this grant, and what is the NIH expecting us to do? So we have a lot of very helpful administrative staff. Some of the administrators that we've been working with at IMBC and at the NIH have been so instrumental in organizing documents for us, and I'm very thankful for that. Without them, none of this would have been possible. And so trainees are going to be filling out these standardized progress reports, and then meeting with their mentors, you and me, to discuss their progress and their future plans. And we want to document their progress over time so that it's easier for us to measure how they're doing and to show the NIH that they're making progress as well, because we'll have to create yearly progress reports for the NIH. So the trainees are going to upload all documents to our program's Google site, which I'm going to create shortly. This will have all different resources for the students to download. And the students will present their research at an annual meeting each year. Mostly it's going to be in Hanoi, but hopefully one of these years we can get it in Chicago and bring everybody in for an annual meeting here. Postdoctoral trainees, their measure of success is going to be to return to NIH and IMBC to set up independent research programs. And if they can set up a program that is funded, we'll be providing small startup packages for them, in terms of maybe a $20,000 startup package for them to get their program underway. But hopefully a measure of success for them is them obtaining fellowships and grants, and we'll be all helping them submit for grants to be funded. And I'll work with you, of course. The funding system in Vietnam way better than I do. So I'll be relying on your expertise to mentor them through that. Like I said, as you already have in the past couple of years, or in the past long while mentoring people. And of course we want them to be productive, so the NIH will like to see them publishing manuscripts. So hopefully from their work at UIC they'll be able to publish a few manuscripts and use these as preliminary data to acquire other grants. Acquisition of fellowships and independent funding, like I said, will be a measure of success. And Kung and I and one of the trainees will work hard on putting together a proposal to obtain a Bruker Microflex MALDI-TOF mass spectrometer at the IMBC. And of course, another huge measure of success is Trainee 3 bringing back the technology to run consistent in vitro TB screening assays there. So that's a major part of the grant. Predoctoral trainees will hopefully obtain jobs once they graduate it's somewhat related to drug discovery. A creation of a strain library from our collection trips is going to be a measure of success, and also the successful carrying out of these dissertation projects that we're going to set up. Hopefully the predoctoral trainees are going to publish several manuscripts each in international peer-reviewed journals, and they themselves will also acquire fellowships. And them presenting at conferences will also be very important. So yeah, with that, these are all the people I'd like to acknowledge, of course, all the mentors who agreed from UIC. Kim Huang and Elizabeth Ryan are our administrative staff who have been just incredible. Kung, who has just been the best partner and collaborator somebody could ask for, and his very strong team here that I actually really look forward to working with in the next couple of years. And Professor Dang, at the NIH, who again is I haven't been able to work with too long, but in the very short period I've worked with him, it's been an incredible honor to communicate and with such a prominent internationally known scientist and his team here, and especially Hung Timai Tran, who is at all hours of the night has been responsive to our requests for information. So yeah, thank you to all of you, and I really look forward to visiting you in person and meeting with you and just answering any questions you may have. But your expertise is and your hard work is what made this grant possible. So thank you very much to all of you. And that's it. I think hopefully I will see you in Hanoi very soon, maybe by the end of the year.